Welcome, everyone, to the SADS Foundation webinar on gene therapy and arrhythmogenic cardiomyopathy. I'm Marcia Baker, the program director for the SADS Foundation. We are pleased to offer this educational webinar with the support and sponsorship from Tenaya Therapeutics and thank Jeff Harrelson of Tenaya for his collaboration. In addition to Tenaya Therapeutics, I also thank our speakers, Dr. Parikh and Dr. Pullman, who are volunteering their time and expertise for this SADS Foundation webinar. It's now my pleasure to introduce our speakers. First, we'll hear from Dr. Victoria Parikh, who is a clinician scientist and assistant professor of cardiovascular medicine at Stanford Medicine. Dr. Parikh's clinical practice and laboratory is focused on the genetics of cardiomyopathies and their associated arrhythmogenic substrates. Following Dr. Parikh, we'll hear from Dr. Matthew Pullman. Dr. Pullman is currently Senior Vice President, Clinical Development with Tenaya Therapeutics. A cardiologist and molecular biologist by training, Matt has served in a number of medical and scientific roles with different corporations, as well as served on faculty at both Harvard Medical School and Morehouse School of Medicine. Following both presentations, we'll have time for question and answer. You may post your question in the chat and we'll address them at the end. Now I'd like to turn this presentation over to Vicki to share her screen and get us started. Vicki.
Thank you so much, Marcia. It's really nice to see everybody here. I see some familiar names out there, Sean and Wendy and everyone else from Tenaya. I'm just going to get our set up here. So it's really, really great to be here. And I really appreciate the invitation from the SADS Foundation and from Jeff. And it's really a pleasure to be speaking with someone as experienced as Dr. Pullman. So I, as Marcia mentioned, I'm at Stanford and I specialize in the care of patients with arrhythmogenic cardiomyopathies. I am also the director of the Stanford Center for Inherited Cardiovascular Disease, which has been around for about 15 years now and sees patients with all sorts of genetic heart disease from familial lipidemias and hypercholesterolemias all the way up through arrhythmogenic cardiomyopathy. So I'll be talking today from the perspective both of a clinician and from a scientist and sort of thinking about how we can put together our understanding of arrhythmogenic cardiomyopathy with the potential novel therapies that are coming out of several companies, including Tenaya. So my disclosures include consulting for Lexeo Therapeutics, Biomarin, and Nuevacor, as well as Viz.ai. And I also have sponsored research from Biomarin Pharmaceuticals. And that research is not related to today's presentation. So what is arrhythmogenic cardiomyopathy? I want to give an example of a gene that that was initially, I think, thought to be a cause of a RVC, which is desmoplakin. And the reason that I want to give that example is that we know that PKP2 or plakophilin-2, which we're going to be talking about a little bit later, and which Dr. Pullman will talk about extensively, is a major cause of a RVC or arrhythmogenic right ventricular cardiomyopathy. But it turns out that that specific definition of a RVC, I think we learned over time was fairly limiting with respect to the spectrum of cardiomyopathies that we can see in conjunction with a genetic diagnosis that causes arrhythmogenic cardiomyopathies. So DSP or desmoplakin is an example of that evolution of our understanding. We first really started to recognize this as a cause of arrhythmogenic cardiomyopathy, a RVC, in 2002 when this paper that I'm showing here was first published reporting that association. And then, for the next 6 years or so, really focused on this as an ARVC gene. In 2008, however, this entity called ALVC was first reported by Bill McKenna's group, which was the same sort of fibrofatty infiltration of the left ventricular wall rather than the right ventricular wall. And as you can see, many of the patients that were sequenced associated with this phenotype actually did have variants in desmoplakin. In 2015, I think that we started to recognize that Perhaps DSP had a slightly different trajectory for patients than those with PKP2 cardiomyopathy, for example, in ARVC. And then most recently, in 2020, Adam Helms and his group led an international collaboration to try to understand really the phenotype of desmoplaque in arrhythmogenic cardiomyopathy, and found that overall it really is much more likely to present with left ventricular cardiomyopathy findings, and that the LV function is actually more indicative of outcomes than RV. And so I bring this up as an example of a problem, which is that now we really have multiple definitions of what arrhythmogenic cardiomyopathy is. On the left, we have probably the broadest definition, which was published in Heart Rhythm in 2019, led by Dr. Tobin, really to try to say, look, let's think about this very clinically. If a patient presents, their first presentation is with arrhythmia, then how can we tell if they might have an arrhythmogenic cardiomyopathy that has a genetic underpinning? Where if that's the case and you, present first with clinical arrhythmia or ventricular arrhythmia, and then find that there's ventricular dysfunction, then you're thinking about testing for those genetic causes. And then there's another approach, which is really to focus on the imaging itself. And that is to say, is this a left dominant fibrofatty infiltration with a specific sort of pattern of fibrosis in the LV, or is it in the RV, or is it in both? And if it doesn't fit those specific imaging criteria, then we don't think of it as an arrhythmogenic cardiomyopathy. So I think that, these are two approaches to the question, but when I think about patients, I really think about the question of how do we, for the patient in front of us in clinic, how do we best help them? What is most clinically useful? I think about their natural history when they have a genetic cardiomyopathy. They, of course, inherit a causative variant before birth. They then may develop symptoms or incidental imaging findings, which lead to diagnosis. And then their life will continue. And either it will continue on a trajectory that's toward an unfortunate and unexpected event like sudden cardiac death, or needing to progress to advanced therapies for heart failure, or they'll live a very happy long life, maybe with some medication, and will pass away from other causes. So when we think about where we can intervene in this natural history, I think we think about getting the genetic diagnosis and being able then to think about disease prevention. And then we think about disease modifying therapies. And so when we define those two moments in a patient's life, I think we know that our precision diagnosis really informs the choice of precision therapy. So if we have a genetic diagnosis, we then can modify that gene's expression or that gene's activity in order to change the course of that patient's life. Of course, we also know that genetics can help us find patients at risk of life-threatening arrhythmias before they have those events. Cindy James has done a ton of work on this with PKP2 and other ARVC genes. The important point here is that overall, in order to be as clinically effective as possible with arrhythmogenic cardiomyopathies, genetics, I think, are really critical to our understanding of patient prognosis and to the development of precision therapies. So we know many genes that are involved in ACM. Over here was sort of the original place in the cardiomyocyte where we identified these in the desmosome. You can see plakophilin 2 here, which of course is responsible for a significant majority of true ARVC by those imaging criteria, as well as other electrophysiologic criteria. There's desmoplakin, which we talked about. And then there are several other components of this complex, which is important for cell-cell adhesion and keeping those cardiomyocytes in contact and communication with each other. There are other genes that have been associated with a particularly arrhythmogenic presentation. For example, laminopathies, or the lamin AC gene, which makes up part of the nuclear envelope, is known to cause a really progressive and highly arrhythmogenic left-sided cardiomyopathy. FLNC, or filament C, same thing. And then we've done some work as well on RBM20, which actually acts completely differently than any of these other structural genes to change the splicing of other transcripts in the cardiomyocyte in order to modify both arrhythmogenicity as well as contractile function. So what do we know about the modus operandi of variants in these genes that actually lead to disease? A lot of this work has been done by Faraz Sheikh, who's a sort of central figure I'm showing here on the basic science side. And then also, this is an example of some work by Roddy Walsh, sort of emblematic of the group from London that really has done a lot of this amazing population-based genetics work to try to understand what types of variants cause disease in these genes. What I'll point out to you is that in that population work, this ARVC work down here, what they're showing is that the farther to the right you are, the more likely it is that the type of variant in that group is actually associated with ARVC down here. And what you can see is that for both plakophilin 2, as well as desmoplakin, as well as desmoglein and desmocolin, that lots of function variants, which are here in blue, are actually even farther to the right than other missense variants that are not actually causing a truncation and early stop in that gene's transcript or protein. And then moving over here, what we know about the missense variants or non-truncating variants that have been shown to be associated with arrhythmogenic cardiomyopathy, both in plakophilin 2 and in DSP from Dr. Sheikh's work, is that it's actually an instability of that protein because of that missense variant, which leads to a loss of function or haploinsufficiency mechanism. So this is important because it tells us that if we could actually restore the expression of that protein in the myocardium for PKP2 or desmoplakin, or potentially also desmocolin or desmoglein, we might actually be able to help repair that myocardium, or in some cases, if early enough, even prevent onset of ARVC. The question then from a clinical perspective is how you detect the right patient who will definitely get ARVC in the future. So we have done a little bit of work on this as well, but I want to show you here that the sort of basic science underlying the idea that there's loss of function in plakophilin 2 leading to disease shows that you get basically, because of that destruction of the desmosome, it falls apart. You get actually a destruction of the overall stretch sensing capability of the cardiomyocyte. There's also some signal for Wnt signaling and other sort of pro-adipose signaling being deranged downstream of that PKP2 disruption. You can also see here that as this whole apparatus is connected to the nucleus, you do actually see that the nuclear envelope gets disrupted in these cardiomyocytes as well. And then when you look at the desmosome itself, you can see that there is disordered desmosomal structure in plakophilin 2 loss of function. So, as I mentioned, missense variants are also associated with disease. We know from prior work in desmoplakin and PKP2, as I mentioned to you, that missense variants can cause haploinsufficiency by causing a loss of function in the protein. But I do want to mention that there are also missense variants, for example, in RBM20, which we've shown in our work to be associated with arrhythmogenic cardiomyopathy, that in fact cause disease via a dominant negative mechanism. So we know that in RBM20, as I mentioned to you, it's a transcription factor. Missense variants in RBM20 that cause mislocalization from the nucleus actually end up causing what we believe to be a more dominant negative form of arrhythmogenic cardiomyopathy, which then may not actually be treated by replacement of RBM20 in its wild type form. Of course, we've done a little bit of work to try to understand where those missense variants are that cause disease. We've done this with population databases, and we know that this arginine serine-rich domain of RBM20 is important for that nuclear localization, which means that those variants in particular, we may have to think about other strategies other than gene replacement, as I mentioned, for our desmosomal genes to actually treat that disease. We have some preliminary evidence, as has been shown by others, that variants in this part of the gene actually cause haploinsufficiency. And so it may be that we don't have a single answer per gene when we think about the ways that we need to treat these diseases from a precision perspective. As I mentioned, we've done similar work in plakophilin 2. This is actually looking at the ratio of missense variants in the disease population in blue to the general population, which we assume doesn't have a RVC. And we see that there are actually several regions which are highlighted in light blue where there are more missense variants detected in the disease population, indicating to us that these may be regions where missense variation is not tolerated by PKP2 and it might cause protein instability. So we're chasing that down as we speak in the lab. So back to this idea of precision therapy and where we can act and how we can develop those precision therapies based on a genetic diagnosis. I think there are sort of two major modalities that we think of when we think about precision therapy. Certainly there have been small molecules developed, which have been aiming sort of at the downstream consequences of specific genetic variants. For example, there was a trial of a novel small molecule for the treatment of Lamin cardiomyopathy, which unfortunately was stopped in phase 3 trials. But really an attractive approach here has been the idea that, well, we know what the problem is, especially for these haploinsufficient variants. And so why don't we actually just try to deliver a good copy of this gene to overcome the loss of one of the copies in our patient? There are several ways to do that. Most successful delivery to the heart is through what's called an adeno-associated virus. These viruses are not infectious in that they cannot multiply. They lack the gene to actually make more virus, but they can be used as a delivery mechanism, like a package that will go to a specific tissue. And so there's a specific serotype or several specific serotypes of AAV9, which will target the heart, and they can carry, as I mentioned, a good copy or a wild type copy of the gene that is missing in that myocardium. We'll talk about some approaches to PKP2 in a moment that have been used, and of course, in the second half of this program, we'll hear much more about the clinical programs that are targeted at those therapies. There are also other delivery mechanisms underway, which include lipid nanoparticles like the one that's shown here. At present, most lipid nanoparticles get sort of chewed up by the liver. And so one, major thrust of research right now is trying to figure out a way to target those lipid nanoparticles and get them taken up into the myocardium specifically so that we can actually use them to deliver transcripts or other machinery to the heart to be able to treat it more precisely. And then down here, I'm just showing you a picture of a very special molecule. So you may have heard of CRISPR-Cas9, which was one of the 2 Nobel laureates came out of the discovery of that machinery. That machinery is very special because it actually breaks the genome and causes an edit. So you can change or rewrite the sequence of the genome. That initial sort of generation of CRISPR-Cas9 came along with a bunch of issues around, well, am I making a bad break that's not getting fixed, and what's my efficiency, et cetera. And from there, some really brilliant scientists have grown out many different sort of iterations of this that can just change base chemistry rather than actually breaking the DNA. Or in this case, this is called prime editing, that was discovered by David Liu in addition to many versions of base editors that can actually rewrite a segment of the DNA rather than just changing one base at a time. And so I think part of the dream will be for those diseases that are not due to haploinsufficiency or are due to haploinsufficiency of a gene that's just too large to fit inside of the particles that we currently have available for delivery to the heart, that we may actually be able to package the machinery necessary for rewriting the genome, as it were, with these CRISPR-based derivatives. So I want to talk now briefly just about 3 different trials, or rather preclinical studies that came out at the end of last year and earlier this year. These are each examples of how gene replacement therapy can actually be used to treat plakophilin 2 cardiomyopathy and even prevent it in a mouse. Each one of them carried a different, developed copy of the plakophilin 2 wild type construct under a different promoter, meaning that's the piece of DNA that controls expression of that downstream construct in an AAV capsid. And so each one of these used an AAV mediated delivery system. I think each had a slightly different AAV for delivery, packaged PKP2 and delivered it to a different model of loss of function of PKP2. One of these studies used a knockout mouse, another used a variant that was engineered, and another was a variant that was engineered from a patient. And in all three of these studies, there was actually quite a bit of success in curing these mice and curing them of their disease. I'm going to talk about one of those studies, and this is really exemplary, I think, of the results that we're seeing in all three. Very exciting. In this particular study, these mice that had a loss of function variant in PKP2 had this AAV delivered retroorbitally at 4 weeks of age, which is pretty young for a mouse. And then their hearts were taken at 6 weeks. And what we know is that if you take, or what we found out, not we, but the authors found out, was that the control mouse, which was a wild type mouse, had a heart that looked like this on MRI. So here is the left ventricle, and here is the right ventricle kind of wrapping around it, just like you might have seen if you're a patient on your own MRI, and just as we all see daily in our own clinics. What you can see is that when you have a homozygous PKP2 variant mouse that has not been delivered any gene, but just GFP and the AAV, is that you have this huge RV here, which is consistent with what we see in human ARVC. We see a big blown out RV. And then if you deliver PKP2 in that AAV to that homozygous PKP2 variant mouse, you see recovery of the size of that RV, which is pretty impressive, right? I mean, we're seeing that these animals are not developing disease, likely because they have that PKP2 copy in the mix. Of course, arrhythmogenic cardiomyopathy is arrhythmogenic. The way that this particular paper measured that was by looking at the frequency of PVCs, which are these tall spikes here and our extra beats, as many patients will be aware of, and of course, as many cardiologists hopefully are aware of. And what we see here is that when you deliver PKP2, you don't see those PVCs. In fact, I think in the end, they counted even one PVC as having an arrhythmia, and they couldn't find any of those mice in the homozygous treated group. So I'll just end on a futuristic note, which is what are we going to do about these dominant negative therapies in addition to gene replacement for haploinsufficient diseases like plakophilin 2. This is again that picture of RBM20 mislocalizing outside the nucleus. I think that some work has been done showing that base editors, as I mentioned to you, those change the base chemistry of a gene without actually cutting the genome. Base editors have actually been able to repair a variant in RBM20, which caused that mislocalization. And you can see right here that this is the corrected mouse, and that all of those green dots there that were outside the nucleus have now relocated to the nucleus after base editing therapy. And you can also see recovery actually of the ventricle itself in this mouse with respect to size and function. You know, we are working on understanding where this might be useful in the human genome, and our work basically has been to say, let's find clusters of pathogenic variants that might be not haploinsufficient, but might require correction. And let's see if we could actually rewrite those with a single prime editor so that we might be able to have a single reagent instead of having to develop a new therapy for each person with a dominant negative variant. And what you can see here is that we can create many, many variants with base editors, and then we can use prime editors actually to correct them at rates that are as high as 100% for several variants with a mean of about 60% editing efficiency. So with that, I want to say thank you so much. I think that the future is now as far as gene replacement therapy goes for the genes that are tractable for that particular solution, and that we are continuing to work on solutions for other modalities of cardiac disease. Thank you so much.
Thank you, Vicki. And now we'll turn it over to Matt to share his screen.
Hopefully it's all a go here.
Looks good.
Okay, thank you. Thank you, Marcia, and thank you to the SADS Foundation for hosting this this evening. It certainly is a privilege to be here with you, and looking forward to the discussion and interaction after this presentation. I certainly want to thank Dr. Parikh for that outstanding presentation. I think as you will see, it will set up very nicely this next discussion that we'll have on the development of AAV-based gene therapy for PKP2 positive ARVC. So I'm Matt Pullman. I'm the Senior Vice President for Clinical Development, and in that capacity, lead clinical development for Tenaya Therapeutics. So here's what we'll try to cover in the next 20 minutes or so. I'd like to briefly introduce you to Tenaya Therapeutics. Who are we? What do we stand for? What do we do? To build off of that excellent foundation that Dr. Parikh laid for us to discuss the basics of AAV gene therapy, particularly as it relates to the approach that Tenaya is taking to address this disease state with AAV9. And then to introduce to you the RIDGE-1 study, which is a phase 1b gene therapy clinical study for ARVC that is caused by mutations in the PKP2 gene. So who is Tenaya and what do we stand for? I think I want to start here with kind of our core value and our core mission, and that is patients first, to be able to transform and extend lives through the discovery, the development, and eventually the delivery of potentially curative therapies that target the underlying causes of heart disease. So we feel that what we have built here over the past roughly 6 years is a company with capabilities and capacities to be well positioned to lead the innovation in precision medicine that Dr. Parikh introduced. Now is the time. We're in a new era. It used to be when we discussed genetic testing, one of the pushbacks from a very pragmatic standpoint was, well, then what? So I know the genetics, I know the genetic cause of the disease, but how does that change what we do? Well, hopefully we'll be able to discuss over these next few minutes. This is now the then what. And we're not alone in this effort, and I'll introduce you to some of the other efforts, but now is the time to be able to implement a precision-based medicine based upon specific genetics. So we are a company of about 100 individuals based in the San Francisco Bay Area. We are singularly focused on heart disease. And what we'll be discussing later in this presentation is one of our 3 clinical stage programs. And that, of course, is what we term the TN-401 for PKP2-associated ARVC. In addition to that, we have 9 early stage programs, again, all focused on heart disease. As part of the capabilities that we've built over time and the systematic approach that we've taken, is that we've screened over 1 billion novel AAV capsids and regulatory elements, which are really the 2 elements that really drive the specificity of the cell type to which an AAV has an affinity, and how well that particular gene is expressed within that particular cell type. So we've been very systematic, and I'll share with you the choices that we have made. And in addition to that, a very critical component of this type of an approach is your ability to consistently manufacture at larger scales the type of material, this AAV-based material that is needed for these types of clinical trials. So as I shared, we're actually not alone in this endeavor. If you look across other indications and other fields, there's actually been a growing body of clinical experience with AAV-based approaches, and now approaching over 20 years. So if you look at the big picture, there's been about 5,500 patients that have been treated There are actually 8 FDA-approved gene therapies, and there's up to over 15 years of follow-up as it relates to really understanding any of the long-term safety issues, and so far it is looking very good. Of course, it is new largely to most of us in the cardiovascular and cardiomyopathy field. But again, if you look across the spectrum, there are about 13 AAV gene therapy trials that are currently underway in the cardiovascular area and cardiomyopathies. So just to review a bit, I know that Dr. Parikh spent some time helping us understand what are the approaches to gene therapy. And they fall really roughly across 2 broad categories, whether that's at the using and manipulating at the DNA level or at the RNA level. We'll spend the focus of our time in gene replacement or gene transfer, where you actually deliver a working copy of the gene, frequently using viral delivery vectors, and we'll talk about those specifically. And of course, this is best utilized in situations in which the mutation causes a loss of function, as described earlier. And the approach would be to reintroduce a normal working copy to compensate for that loss of function from the mutated gene and mutated protein. Dr. Parikh also introduced gene editing. Again, we won't spend much time on that, but that's an approach where you actually change the cellular genomic content, as she discussed. And of course, at the RNA level, you can silence genes in which the protein that is produced is actually somewhat of a poison peptide and is actually disease-causing by its presence. So you want to knock that down. So those are kind of the high-level approaches to gene therapy, and we'll spend the remainder of our time on gene transfer or replacement. So let's talk about AAV. It certainly has become the delivery vehicle of choice as it relates to delivering these types of therapies. And there are a number of reasons why it is particularly well suited for these types of approaches. It has many appealing features as a vector for gene therapy delivery. The genome itself can be easily removed and replaced. And of course, that's what we're doing at Tenaya with a desired transgene, a therapeutic gene. In this case, it's PKP2. The various AAV serotypes and capsids have broad tissue tropism, and they're fairly effective. These really were designed over time in nature to be very effective at infecting and transducing certain cell types and be very efficient at introducing genetic material into mammalian cells. In essence, we're just trying to harness that capability and transform it into something that is actually therapeutic. The good thing, of course, is that these recombinant AAVs deliver genes without integration into the genome. So we're not worried or concerned about the risk of insertional mutagenesis. And it also has a long-term persistent episomal expression in non-dividing cells, which of course, in our case, the target cell are cardiomyocytes, which do not divide. And hence, there's that opportunity for that episomal to have long-term expression of the therapeutic gene. So let's drill down a little bit further as to how this all happens. What are some of the key components of recombinant AAV? As we discussed, the most simple approach is to take a wild-type AAV, which in our case, we are using a wild-type AAV9, and you delete out the native intrinsic genes to the wild-type AAV, and you reinsert genetic material, which includes really 2 key components here: a promoter, which is a DNA sequence that controls the gene expression by initiating transcription of a gene. And for the most part, these are very tissue and cell type specific, which of course means that no matter what cell type the recombinant AAV may have been transduced into, the actual transgene, in this case PKP2, would only largely be expressed in this case, cardiomyocytes and not other cell types. Of course, the transgene is the therapeutic itself, and that is introduced along with this promoter. There are some practical considerations. I know that Dr. Parikh mentioned the DSP gene, and the condition, of course, has a much more severe course in terms of its clinical presentation than PKP2. However, there's a practical consideration in that there's a limit to the packaging that you can put into a recombinant AAV. Largely, the transgene size needs to be about 4 kb, and in the case of DSP, it exceeds that, and so it really is not a good candidate for this approach. And Dr. Parikh described other approaches that are on the horizon or in early clinical trials. A real key component, of course, of this approach is the actual capsid itself. The capsid really determines the cell type that a particular AAV has an affinity for based upon what is expressed on its surface, this protein shell, and the specific receptors that are unique to many cell types. And it's trying to match that combination of the type of proteins that are expressed on the surface, along with the types of specific receptors that are expressed on cell types, and that's how you develop and can control some degree of specificity for the therapy as it relates to a specific cell type. And as you look across the serotypes of AAVs, there are a number of them here listed in this table. If you look down to AAV9, this is the particular serotype that we have chosen to work with, and largely because it has a specificity for heart, and in particular the cardiomyocyte, which in the case of this disease state would be the target cell. And so has the optimal opportunity here to be more specific for the heart tissue and the condition we're dealing with than with other off-target cell types and tissues. So to describe a little bit further some of the advantages to going with AAV9, it does come with rather robust validation. It does have the most well-established safety record of any capsid, been used in over 4,000 patients in a number of countries with over 8 years of follow-up, and the majority of those being the treatment of Zolgensma for SMA. And it does happen to be, at this point, the only capsid with validation from human hearts for the biodistribution, transduction, and durable expression. In this case, it's the Danon disease, and it does have a very extensive body of preclinical evidence in disease models, and I'll share some of our own data with you. And it's proven to achieve high expression in cardiomyocytes, and I'll share some of that data with you as well. So, and you take that in totality, it certainly becomes a very compelling choice, particularly for this disease state. As we've shared with you, we're not alone in this endeavor. There are 2 other sponsors that are approaching PKP2 associated ARVC with a gene replacement approach. However, we do differ in the particular capsids that we're moving forward. I described to you the reasons why we chose AAV9, and because of that level of specificity for the cardiomyocyte and some of the work that we've done on the promoters, we have near maximal efficacy at a fairly low dose. For those of you who are new to the field, certainly staying in the lower E13 range is desirable from a dosing perspective. It also lessens the chance that you'll get some of the off-target effects or some of the side effects and toxic effects that have been seen in rare circumstances, but usually at much higher doses. So this allows us to start at a clinical dose of 3E13, and as you'll see in the trial design, to escalate by doubling that dose in the second cohort to 6E13. So what's the direct evidence that AAV9 does outperform the other two capsids? This is data that was recently presented at the ASGCT 27th annual meetings. If you look at this, this is an in vivo study in which we married a reporter gene, in this case GFP, with a cardiomyocyte-specific promoter. And again, that means that this particular reporter gene would not be expressed in other cell types, except if the transduction and expression occurred within a cardiomyocyte. So after constructing these three therapeutic AAVs, we injected them into mice, took out the heart tissue, and then took measurement of how effective was each of these in transducing or getting into heart tissue, and then looking specifically at what was the transgene expression at the RNA level, and then finally, how did that translate into protein levels in the heart? So if you look at panel 1 on the bottom, actually all three of these were fairly effective at transducing heart tissue. But again, if you remember, the heart itself is composed of a number of cell types, and cardiomyocytes in and of themselves really only represent 30 to 35% of the cell type, although by mass or volume, it's about double that. But nonetheless, certainly there's that opportunity to transduce other cell types in addition to cardiomyocytes. So what we see, based upon the specificity of expression given that cardiac promoter in panel 2, is that AAV9 in the blue bar outperformed the other two capsid almost by threefold as it relates to RNA expression. And furthermore, that translates into a roughly threefold expression level as compared to the other two capsids of the actual protein. So that gave us confidence that based upon all the screening work that we had done, that we had indeed chosen a capsid that was going to be fairly effective in its specificity for cardiomyocytes and our ability to express the RNA and protein in the cell type and target cell type of choice, and that is the cardiomyocyte. So I won't spend too much time reintroducing you to PKP2-associated ARVC. Dr. Parikh did a wonderful job of introducing the disease state. But again, to remind you that at the foundation and initiation of the disease is a lack of the expression of one of the normal copies of the PKP2 gene. And again, that is a critical component of the desmosome, which is associated with structural integrity as well as electrical stability or instability. So to put it all together, in essence, what we've done is remove the viral DNA from AAV9. We've inserted a normal working copy of the PKP2 gene. We would administer this as a one-time intravenous infusion, where, as I've demonstrated to you in the preclinical studies, that TN-401 effectively targets cardiomyocytes, where it delivers a working copy of the PKP2 gene. And then the goal, of course, would be to restore functional protein and to modify the disease. So that's the overall approach that we're taking with TN-401. Well, what gave us confidence that this has an opportunity to work in the clinic? We certainly utilized some of the best preclinical models that we could find. And in utilizing that same approach with some modifications for the size of the actual mouse, we used a model in which there's a conditional knockout in the heart of the PKP2 expression. It leads, in the case of the blue bar, which is the CKO, or the conditional knockout, a very high incident of arrhythmias. It markedly reduced the ejection fraction and leads to RV enlargement. As we reintroduce using the approach that I just described, the TN401 and PKP2 gene with AAV9, we can see that in a dose-dependent fashion, we get reduction in the arrhythmias, we improve the ejection fraction, and we effectively reduce the RV enlargement, and we're able to do that at doses of as low as 3E13, and that will be the justification for our clinical starting dose. So where are we on our clinical development plan? The great news is that we have cleared the IND. We are actually activating sites in our RIDGE-1 study, which will eventually become a global phase 1b study for safety and proof of activity. And site activation is currently underway, and we have 4 sites that are already active, and we'll begin screening patients for enrollment. What has helped to generate our understanding of the patient population that might be most eligible for and most likely to benefit from the gene therapy is our global phase 0 study, which is essentially a registry of PKP2 ARVC patients, where we study the Shero status, and I'll get to that in a minute, as well as some baseline and key eligibility criteria. And we'll talk a little bit about more about that here in a couple of slides. So one of the drivers for this study was to better characterize neutralizing antibodies that may be present in individuals who are interested in entering this study. While AAV does not necessarily cause disease, it can, infection with AAV does mount an intrinsic antibody response, an immune response. If you have been exposed and have a high enough level of response, these neutralizing antibodies could actually prevent the entry of the capsid into cells, or could reduce the transduction and treatment efficacy. So there have been in vitro assays developed that can measure the level of the neutralizing antibodies that are present. That's one of the measurements that we are taking in our phase 0 study. So to date, our RIDGE, or phase 0 study, has enrolled over 70 patients across 3 countries. And the good news is about 81% of the enrolled participants had a titer that was less and was eligible then for the criteria that we set for the phase 1b study. And there appeared to be no differences across age, gender, or geographic region. To date, we have 16 sites that are enrolling in this phase 0 study, and with 7 additional sites that are pending activation. So we've done some characterization. Certainly it was critical to have the right variant, whether it needed to be pathogenic or likely pathogenic for PKP2. As expected, most of these do not have New York Heart Association functional class beyond really 1. About 63% of them have no symptoms. What is important is that we characterize some evidence of electrical instability as evidence of some progression of disease. And so an elevated PVC count in this case will be important, and important criteria for entry into the study. And so we've been characterizing that. And as you can see, the median here is about over 2,000 in the period of 24 hours. So here's the slide that describes the phase 1b clinical trial. Our IND is cleared, as I shared with you, and the hope is to begin dosing in the second half of this year. So just to run through very quickly, eligibility, obviously need to have ARVC, the appropriate mutation. This is an adult study. They need an ICD. And then part of the reason we're characterizing the PVC count in the phase 0 is that these individuals do need to have an elevated PVC count to be eligible for entry. There needs to be some evidence of electrical instability, and essentially some target that we can follow, some dynamic marker that we can follow as a response to therapy. So this is an open label, multi-center dose escalation and dose expansion study. There will be a preventive immunosuppressive regimen and close safety monitoring as part of the study design, and regular assessments for safety, PK, and PD, and overall 5-year safety and efficacy follow-up. The goal is to enroll 2 cohorts, with cohort 1 at the lower dose of 3E13, which I shared with you, was near maximal in terms of its efficacious, the response in the mouse model, and patients will be dosed sequentially in a sentinel manner. They'll undergo a safety review after the first cohort is dosed, and then be able to proceed to the second cohort of doubling the dose with, again, a review. So the obvious endpoint here initially is safety and kind of feasibility and tolerability, with, again, a very important couple of readouts. We will be taking cardiac biopsies at baseline to establish the level of PKP2 protein expression at baseline. And then we'll be looking at 8 weeks and 52 weeks, where we'll, in addition to looking at mRNA and protein expression, we'll be able to tell the transgene expression and the effectiveness of the transduction, particularly at that early time point at 8 weeks. We need to be effective at getting the AAV9 to the target tissue. And then over time, we would expect to see increases in the transgene and mRNA expression and the protein expression. So the goal would also to be to be able to see some change in what we would call really our pharmacodynamic markers. And in this case, we're looking at PVCs and non-sustained VT, with a number of also exploratory efficacy endpoints. So this gives you a visual representation of the clinical trial sites and the state and where they're located. Again, we're looking at roughly 7 sites that will be activated, and hopefully fairly broad geographic distribution here across the United States. And we also intend to open sites overseas, initially in the UK. So with that, that's the close of the presentation. I want to make sure that you have the information that you may need in case you want to reach out and contact Tanaya further. And I'm sure that also the SADS Foundation will be able to point you in the right direction should you have any additional follow-on questions. But with that, I'll pause in the presentation, and I think we'll transition to a Q&A, but I'll turn it back over to Marcia.
Thank you very much, Dr. Parikh and Dr. Pullman, for those excellent presentations. Now, if anyone on our Zoom call has a question for either physician, please type it in the chat and we'll address them as we go. Matt, do you want to go ahead and you can stop sharing your screen? And we can see. Oh, there you go. Okay, a question from Yuki Asaki. Are there clinical characteristics that would make a patient too sick to enroll?
Yeah, no, that's a great question. Something that we spent a fair amount of time discussing with our advisors and thinking through very carefully. Certainly, the initial studies, we want to make sure that we're not selecting individuals whose disease progression has passed to a point and progressed to a point where it would be difficult to demonstrate a therapeutic effect. In this case, we are obviously concerned about very progressive fibrofatty replacement in the right ventricle. The thought would be it would be very difficult, we think, to we demonstrated to you we actually do need active and healthy cardiomyocytes to transduce and to be able to express PKP2 protein and restore function. So obviously, if you have a tremendous amount of fibrofatty replacement, you don't have those myocytes to transduce, and so the opportunity for therapeutic benefit would seem quite slim. So if you look at very severe RV dysfunction, severe dilatation, those patients we would exclude from these initial studies for the concerns that I outlined.
Okay, thank you, Dr. Pullman. What are the possible side effects?
Sure, another great question. So what we know, and this is across any of the studies that use AAV, and certainly we know a lot about AAV9, is that there is an initial innate immune response to the virus, and predominantly the virus capsid itself, and predominantly affecting the liver. The liver does a great job of filtration, and it does a very effective job with the AAV. There's a lot of particles that eventually get trapped in the liver. So there is an immune response. In many studies, there have been maybe up to 80% of individuals who receive the therapy that have elevated liver function test, transaminase. These are, for the most part, quite treatable and monitorable. And in the case of our study, we'll be using immunosuppression, a combination of steroids and Alimus in order to control that response. But nonetheless, there is most likely going to be some elevation of LFTs. Something that's quite rare, but can be quite serious, is thrombotic microangiopathy, or TMA. If you look at studies that have tracked the safety of AAV9 in longer-term studies, this is about less than 1%. It does tend to occur at higher doses exceeding the E14 level. And again, as you saw in this particular study design, we're trying to stay well below that. This, again, is monitorable and treatable. And it's really all about complement activation. And there are therapies to reduce the levels of complement activation as well. And certainly that's something that we will be monitoring in our study. So I think those are LFTs being the most common, TMA probably being the most serious. There can be a reduction in platelet counts, thrombocytopenia. Again, that tends to be transient, treatable. And for the most part, not serious unless that's actually part of the TMA syndrome itself.
Thank you. If patients participate in phase 1, will they be eligible to enroll in subsequent phases? And then just kind of a counter to that, if people don't participate in phase 1, can they participate in a later phase?
Sure. So, just as I described, the monitoring that we're doing and assessment that we're doing for neutralizing antibodies to exposure to AAV9, Certainly, if you participate in the trial, patients participate in the trial, receive a very large dose of AAV9, their body will respond to that. So at this point, while there are technologies that are in development to potentially suppress a response that exists or pre-existing response. Currently, an individual would be ineligible to receive another dose of an AAV, and particularly AAV9, simply due to the presence of high levels of neutralizing antibodies. Of course, if you don't participate in phase one, absolutely, an individual could be eligible for later studies. And not participating in phase one certainly does not make you ineligible for phase two. Also want to highlight the fact that if you don't participate in phase zero, which again is this registry study and serostatus study, that also does not mean you cannot participate in phase 1B or subsequent studies.
Thank you. So let's see, does a patient have to be off their medication while being evaluated? Their sons are on nadolol and protoprolol, which control their arrhythmias. So on medication, they wouldn't have 500 within a 24-hour period.
Yeah, obviously, any modification of your medication and the adjustments are really between the patient and the treating physician. We are not recommending any specific changes. I think the individual should be on standard of care, and again, that standard of care being best determined by the physician and the patient. I think if you look at some drugs and therapies that individuals may be on, that would be a challenge for the study. Certainly amiodarone, given the potential for liver toxicity that exists for amiodarone, I think that's a challenging one. But again, we're not asking that physicians make adjustments to medications. It's patients first, whatever is the most appropriate therapy for an individual. We'll just take it as it comes, and we'll look for eligibility criteria that with the patient being on standard of care.
Okay, thank you. I think you answered this one. Do patients need to have an ICD to participate?
They do. Yeah, they need a functioning ICD with remote interrogation capabilities.
Okay, thank you. Any other questions from any participants? Okay. Are there other heart diseases besides having ARVC that would exclude patients and or like blood disorders? And if you've been on amio and have toxic side effects, is this not a good trial? Sean.
Yeah, I think, as I said, amiodarone in particular is a challenge. I think we would want to avoid any other major cardiac disease, and certainly any other associated disease that would be arrhythmogenic. And I think that we need to just look at each patient case by case.
Okay, thank you. Any other questions? Okay. I have one that's been sent to me, and here's another one. How long would patients be sent to a site for treatment?
Or I mean, we can get into some of that. Certainly, there is a hospitalization period for the administration of the AAV therapeutic. And there's very close follow-up for several months, with the most intense period of follow-up being in the first 3 to 4 months, in which the patient is most susceptible and vulnerable to any immune response. And certainly we'll want to get them past that phase and be able to effectively reduce and taper off any of the immunosuppression that they are on. with that being the goal. And I think it does take close monitoring, probably on a weekly basis, in order to make sure that we're able to effectively do that.
Okay, here's another question. What does the follow-up look like if there are liver toxicities? I can't say it. Arise.
Yeah, I mean, the good news is that if you look at the larger studies and some of the registries that have been done with AAV9, particularly with Pfizer and Zolgensma, is that the liver effects are very manageable. Usually, if they're on a steroid taper, you would increase the dose of prednisone. You may adjust any of the other immunosuppressants that they are on, but obviously the steroid is probably the first that you would adjust. And with adjustments of the steroids, there have been very few cases that have led to further liver complications, and for the most part, these are monitorable and treatable and manageable over time, predominantly with adjusting the steroid doses.
Okay, well, thank you very much, Dr. Pullman. Dr. Parikh had to leave, so we appreciate your excellent presentations and addressing our questions, and we thank all the participants for joining us tonight. So this webinar has been recorded, so it will be available to all the other registrants. We had quite a few register for this, quite a lot of interest. So thank you very much and have a good evening.